It's now my pleasure to introduce the next speaker, who is Doug Lindsay. Doug will tell you his story, so I'm not going to take away from his presentation. But Doug is co-chair of our Community Advisory Board for the Institute of Clinical and Translational Sciences and the Institute for Public Health. He's also a member of the PCORI's Rare Disease Community Advisory Board, and has actually acted as a member of community advisory boards for some of the NIH active trials that are addressing treatment of COVID. So I've gotten to know Doug over the last few years. He will bring you, I think, a perspective that is incredibly important, that of the patient as a partner in your research. And so I'll hand over to Doug. Thank you, Doug.
Thank you. I'm going to get the slide clicker here. Well, I wanted to start by acknowledging Washington University cell biology professor Ken Bloomer. For 13 years, he spent time helping me understand how doctors operate, how hospitals work, and how scientists think. The gifts of time and insight and wisdom that he gave to me really mattered. My story was a long shot, to be sure, so his efforts weren't necessarily guaranteed to amount to anything. But without Ken Bloomer, there's no story for me to tell today. So let's see. 1995. This was the year that Pizza Hut released stuffed crust pizza. There it is. That is the cheese baked into the crust. On Seinfeld, Elaine said it will be years before they figure out a new way to stuff more cheese into a pizza. But soon after that came Domino's cheese burst pizza. And this is cheese layered into the crust. So if I'm here to talk about the role of patients and integrating patient input into research, why am I talking about pizza? Well, I think I'm talking about that to point out that it seems legitimate to say that pizza chefs have spent more time and been more adept at figuring out how to integrate cheese into pizza than researchers have been at integrating patient input into their research. So there's a long way to go for us to reach the stuffed crust pizza stage, but I think we have some ideas from some of the work that we've been and from some of the ideas we've heard earlier today to get there. So I'm going to share with you my story, and then I'm going to touch very briefly on 3 other e-patient leaders, because we show the roles that patients are able to play in research already. The roles are extensive, they're just not widespread. So when I was 21, I got sick. Up until the end of my junior year in college, I was just a normal college student living my life. I took a summer research biochemistry position at the University of Kansas, and I attended Rockhurst, which is also in Kansas City. But almost as soon as I took that job at KU, I came down with mono. And I went home and rested for the summer to get ready for senior year. And I made it one day of my senior year. And it was clear that even after months of rest, I wasn't getting better, I was getting worse. And my health deteriorated further. I had to drop out of school, and I became homebound. So in the spring of 1999, I'm a healthy honors student living a normal life and doing some pretty fun things. And by the fall of '99, I'm a grown man laying on the floor of my home, and I'm unable to function. My heart's racing, the room's spinning, I have painful cramping muscles. I could get a heart rate of 140 even while laying down for 2 hours just from eating a peanut butter and jelly sandwich. Sometimes my heart was beating so hard that you could put a piece of loose leaf paper on my chest and watch it flutter. I was exhausted all the time, but rest cured nothing. By my 22nd birthday, it was a challenge for me to change a light bulb. I could reach up and take it out, but I would have to rest before putting the new bulb in because I couldn't keep my limbs away from my body for that long. My life had become a disaster. So I turned to the doctors for help. I saw a series of specialists, and no one could tell me what was wrong or what would help. So even though I was bedbound 22 hours a day and could walk only 50 feet, I decided to take control. I would partner with the doctors, sure, But where we struggled to find answers, I thought I would take the lead and I would tackle my problem like a scientist. So I started reading. This is 1999, 2000. So I'm reading 1,000 and 2,000 page medical texts. I figured I was looking for something rare, because if it was something common, the doctors would have found it. And about a year into my work, I came up with an idea. I theorized that there was likely an entire class of chronic autonomic nervous system disorders. Now people are routinely diagnosed with things like POTS, but at the time, not so much. And so I wrote up what I had, complete with bibliography, and I took it to the doctors, and they said, problems like you described don't exist. And I said, but they could. See, doctors practice from experience. So the idea that a patient has strolled into their office and theorized a class of disorders that they don't routinely encounter or know anyone who specializes in treating sounds absurd. But if you're approaching it like a scientist, the idea that there's a complex system in the body and not a corresponding suite of chronic disorders associated with it, that sounded absurd to me. So that was the stalemate we had. And so for 9 months, that was where things were left. And eventually, I was able to get a computer with internet access and rapidly find that I found a nonprofit devoted to the kinds of problems I theorized existed and was told did not. So as we know, the autonomic nervous system controls heart rate, blood pressure, metabolism, and digestion, among other things. And I had said to the doctors, look, I think if this system malfunctions, we're going to see problems with heart rate, blood pressure, metabolism, and digestion, problems like we were seeing in me. And it turned out, once I started to get access to the literature, that autonomic nervous system problems are called dysautonomias. So now that I had the lexicon of the field and a computer with internet access, my searches became far better. I wasn't searching for horseless carriage anymore. I wasn't searching for sympathetic nervous system dysfunction. I was searching for dysautonomias. So my search results got better, and people like Dr. Bloomer and friends of mine who'd gone on to med school were able to get articles and send them to me. And so 18 months later, I'd come up with something, and I submitted an abstract to present at the American Autonomic Society's annual meeting. So when I explained to Dr. Bloomer that I was getting not a lot of response in my appointments to this idea, he basically said, look, you're talking about the world of research medicine, and there's no front door to the world of research medicine. So thinking about the American Autonomic Society, where all of the researchers from the world, would be gathered who care about this, that seemed like the best front door I could get. So I submitted an abstract outlining a proposed treatment for my own disorder, and I was accepted. The poster before mine was from the Mayo Clinic, and the poster after me was a scientist who'd flown in from Japan. And in the middle was me, a 24-year-old college dropout in a wheelchair, outlining a proposed treatment for my own disorder. The presentation went well, but I hadn't gone just to talk. I'd gone to find a collaborator. And I started making relationships with the doctors in the field that would last a very long time. But I left that conference without a collaborator, and it would take me another 18 months to find one. And I ended up working with Dr. H. Cecil Coughlin. He was the director of the Autonomic Nervous System Laboratory at the University of Alabama, Birmingham. And you can see from this photo, he's engaged. He wanted to work with me. And so that's what we started doing. 5 years into my illness, I'm laying on a mattress in an SUV and being driven down to UAB. I got a proper autonomic evaluation, and it turned out I had a very severe dysautonomia. But Dr. Coughlin's work also showed that there was a chance I had a PHEO. I had a small adrenaline tumor that might be responsible for my symptoms. So if we find it and remove it, I might get better. We spent a year looking for a PHEO, and I tried not to get my hopes up, but it is a long time to be bedbound thinking there's something the size of a marble hidden in your body, and if you find it, maybe you'll get better. But there was no PHEO. So that was one of the hardest times that I faced, is because I wasn't ready to give up, but I didn't know what not giving up would look like. And so I went back to the literature, and I basically asked the question, is there anything that would present with symptoms a lot like a pheochromocytoma, but that wouldn't have been detected by the scans we'd run? And I came up with two things. There was primary hyperepinephrinemia and adrenal medullary hyperplasia. Additional imaging showed that I likely had one of these conditions. And so now the challenge was what to do. Well, first, it's a mouthful. And so when you're talking about on the phone, we sort of shorthanded it to Lindsay Syndrome because Lindsay's my last name. I thought it was catchy, but it's also a rare disease. See, we've had 46 presidents, and I was only able to find 32 documented cases of adrenal medullary hyperplasia worldwide ever, and 5 of the other condition. So statistically, you're more likely to be elected president than you are to be diagnosed with one of these. But if that's what I had, that's what I had. And the good news was it might fit the same model. If we remove the hyperfunctioning tissue, I might get better. So this is an adrenal gland, and the cortex makes steroid hormones that we need to live, and the medulla makes epinephrine. So if we could remove the middle and leave the outside, I would potentially be better. But there was no surgery to do that. There were partial adrenalectomies, but they were a lot like taking a slice of pizza to get, a meatball on it. They were not designed to take the center of the gland and leave the outside. Quan Du at UCSF described trying to take the middle and leave the outside as trying to cut the peanut butter out of a peanut butter sandwich and leave the bread, a nearly impossible task. But it's actually worse than that because the adrenal gland is not the size of a piece of bread. This is my thumb, and that is 11 slices of human adrenal gland. So this is what we would need to take the center out of. But impossible or not, or long shot or not, it was still my best chance to get better. And getting a new disease from the surgery would be a hard sell because we couldn't prove that this surgery would help me, but we knew that if we took both adrenals that I would get a new disease. So if I needed a surgery and one didn't exist, I decided I would invent one. So how do you invent a surgery? Well, I didn't know, but I tried everything and I talked to everyone. I worked with everyone from the chairman of the President's Council on Biomedical Ethics to the Japanese ambassador's son, who was a physician here at WashU. And so he took my records to Tokyo because he could speak medical Japanese and meet with an experienced partial adrenalectomy team there. So for a year, I came up with nothing but dead ends. And then I got a breakthrough, or at least what I thought was a breakthrough. It turned out that the surgery I needed was possible in rats. And so this surgery that we thought was impossible in humans was possible in rats. Now, doctors don't like rat medicine because everything gives them cancer and everything does this, but anatomically, it's a little different. So a basic scientist had done the surgery I needed on rats in 1980 at Georgia State. And so I called him up, and he said he sliced into the adrenal gland with a razor blade and squeezed until the middle popped out like a pimple. Well, when I relayed that to Peter Angelos, head of endocrine surgery at UChicago, he was not impressed. So I kept reading and I kept digging. And it turned out that the surgery I needed had been done in cats at Harvard in 1926 by physiology pioneer Walter Bradford Cannon. Now, Dr. Cannon wrote volubly on so many things, and yet only half a sentence on this. In fact, he spent half a page explaining how he accidentally invented the beer bong, but only half a sentence on the surgery that might save my life. So this was, frustrating. I'd worked with the history of science department there. I'd even talked to Cannon's biographers. They'd looked through things. We couldn't find any more. And I found that the surgery had been done in dogs in Oregon in 1940. And they hadn't explained how to do it either. So this was insane and infuriating. Basic scientists who weren't surgeons 80 years ago thought so little of this procedure that they didn't explain how they did it. But the world's best human surgeons today didn't think it was possible. So we had to keep digging. And a librarian at the Oregon Health Sciences University helped me track down an article from 1923 from Argentina. Now that's the year before The Great Gatsby came out. And it turned out that Bernardo Houssay had done the surgery successfully on 11 dogs. And he explained it like this. He said he sliced into the adrenal gland with a sharp Gillette blade and opened it like a book. Then he scooped out the middle with a small hard spoon and sewed it back up. That was the surgery. People were stunned. So I brought them the info, and we'd shown that this was possible in a 9-ounce rat and a 9-pound cat and a 60-pound dog. And so this seemed to be a scalable endeavor. So now, but the doors didn't just swing open. I brought the information to Dr. Coughlin, and I learned something amazing, but also something illustrative about science. Bernardo Husset developed this surgery in 1923. Dr. Coughlin, who practiced at UAB, was from Chile. And Bernardo Husset's protege was Dr. Coughlin's mentor. And so as he's retiring and leaving medicine, the patient he's been working with for 6 years sent him an article about the Nobel Prize winner he knew personally and worked with a bit, who developed the surgery that might change my life. So we worked for 18 months to pull a team together, and it finally did come together at UAB. And so I traveled to UAB and I got the first surgery, and the surgeon there decided to use the Da Vinci surgical robot instead of a spoon. So that was comforting. I got the first surgery and we didn't know whether it would help me because I'd survived. That was wonderful. But soon my body started to change. And so 3 weeks after the surgery, I could now sit for 3 hours instead of just the length of a meal. And I started being able to walk further. And so I walked further every day. 130 days after the surgery, I'd gone from being able to walk 50 feet on the day of the surgery to being able to walk out of my house and walk 4 miles. So on Christmas Eve 2010, I walked in the snow to midnight mass, and I stood in the back of a church I'd been in just dozens of times growing up, and just tears streaming down my face. And it seemed like a miracle. And my neighbor said, one of my neighbors said to me that a miracle is only a miracle if you can't explain it. Well, okay. After 11 years homebound and bedbound, we took a long forgotten 90-year-old dog surgery and turned it into a modern human procedure that took me from wheelchair to walking. To me, that still sounds like a miracle. So my health plateaued because there were 2 adrenal glands and both had shown up avid for the MIBG tracer. Both showed to be a problem. So I had to go back for the second surgery. And that was its own challenge. But that surgery came together here at Washington University. And we were successfully able to remove medulla and leave a living piece of cortex that's there now. And the recovery from that surgery was challenging. It showed that my hypersensitivity to catecholamines was its own disorder. And so that spurred some of my other work, in which I developed new uses over the course of my condition for 5 old drugs that have been used to treat my family. But eventually, my health got better. I was able to go back to school, finish my degree in biology, and I became a speaker. And so I would travel and I would talk about the role of hope and character and innovation and problem solving. And afterwards, people would always come up and talk to me, and they wouldn't say, like, great speech, come talk to my company. They would say, I want to tell you about my spouse. I want to tell you about my kid. I want to tell you about my neighbor who's sick and stuck. And so speaking is wonderful and I like doing it, but I wondered if there wasn't something more I could do. And so now, you know, I can work with a very small number of people, but people can hire me and I basically join their family and see if we can budge their rare or complex condition and get them sort of over the hurdles that they're facing in the medical system. I think I help, you know, the numbers show I help about 10 times more people than can ever hire me, but that's still a small number. And so one of the things that was most powerful in my journey is that in working with Dr. E.D. Pellegrino, the bioethicist at Georgetown, I saw that he was able to live out an integrated philosophy of medicine. The way he was helping me one-on-one while he was chairman of the President's Council on Biomedical Ethics and finding time to work with me, a long shot hope, you know, college dropout who invented a surgery in his living room, that the way he was able to treat me and interact with me was in accordance with his writings and his philosophy of medicine at all levels of organization, from the individual to the society. And that was so inspiring that it's prompted me to try and do something similar and to do public service work. So I am co-chair of PCORI's Rare Disease Advisory Panel, and I do work with the Institute for Clinical and Translational Sciences here on the Community Advisory Board. And I was happy to work with Active One because when COVID came along, you just wanted to do something. So my study has been amazing, and you know, my story's been amazing, and it's sort of showed the role that patients can play in research. But I'm not alone at all. We don't have time to do justice to these stories, but we can at least tell you that they exist. So if you haven't heard of Dana Lewis, she has type 1 diabetes and she's a heavy sleeper. So she would sleep through the alarms that would come off about her blood sugar. So she got together with some people and developed an algorithm that let her continuous glucose monitor talk to her insulin pump and manage her dosing that way. And there are numerous fail-safes in place. This product has been around for almost 10 years, but she was way ahead of industry in creating, a feedback loop in which the two devices were able to operate together under basic rules that showed clinical success, and rather than having the patient continue to be the intermediary between these two devices. So Emily Kramer-Gollenkoff, I met her at Stanford Medicine X. She has cystic fibrosis. Lots of people have been helped by the recent advances in treating cystic fibrosis, but her cystic fibrosis is caused by a nonsense mutation. And so she's in the tough spot of being in the 10% who are not helped by all the progress that's been made. And so if you understand anything, when 90% of people with a disease are going to get helped, that means that there's going to be a lot less attention paid to the condition, because everyone knows you can't help everyone. And it just means there's going to be a shift of focus. So she and her family started Emily's Entourage, and they've raised $10 million and funded directly research studies aimed at bringing treatments potentially for these nonsense mutation-caused cystic fibrosis cases to market. The third is Sarah Regari, who got her PhD this year. She's over in Sweden. She started having Parkinson's-like symptoms, I think, at 13. And she's become a traditional researcher, but what she has also done is sort of pioneered and been at the forefront of self-science for publication. The kind of tracking she's done is very different than is often done. And so she made this graphic in 2014 to tell a very specific story. To manage her Parkinson's disease, Sarah Regari spends 1 hour in neurological health care and 8,765 hours in self-care per year. From one of her recent publications, conventional group-based clinical research aims to find generalizable answers to clinical or public health questions. The aim of personal science is different, to find meaningful answers that matter first and foremost to an individual with a particular health challenge. The researcher and the participant are the same, which means that specific ethical issues may arise, like protecting the participant against self-harm. To allow patient-led research in the form of personal science to evolve further, the development of a specific ethical framework for self-tracking for personal science is needed. So her efforts to get a PhD were slowed down by years because she was interfacing with an IRB, and she had gathered data throughout her life, long before she was a student, long before she was a researcher. And so now that she's got her doctorate, she has co-authors on this paper, and she has taken this challenge head on, rather than simply pivoting and seeking grants and moving forward. So I'm tremendously proud of the work that she's done. So why do I talk about these folks? There used to be an old saying in NASCAR, and it was win on Sunday, sell on Monday. The car that you would see in the winner's circle at the end of the race, you could go to your showroom and buy. As I've said, patients have had extensive roles in clinical research in different ways, but not broad roles, broadly applicable. And so that's what we're going to talk a little bit about now. I would suggest that we can view subjects, that we can view the participants either as subject matter experts or subjects, and it varies. So patients can play a role at each stage of planning. They can shape experimental design so that it is patient-friendly. They can revise recruitment language so it's clear and readable. They can suggest a process for researchers to check in on participants to see how it's going and avoid participant dropout. And they can help disseminate readable research results, helping to craft the public-facing results, but also sharing the peer-reviewed ones. Now, there is a caveat. Ethically, we need to make sure that including patients does not give them an undue rooting interest in the study's success. The way participants help a study and themselves is by being 100% frank and honest in their reporting to researchers. Being included feels great, and people want treatments for their diseases. And what we don't want is participants to feel a rooting interest, because everyone wants something to work out, and it'd be great to be a part of a win. But we need solid science, and we need results that we can rely on. And so if there are times when ethically it's not appropriate to bring study participants in to help shape things. That's where, if you can't involve enrolled participants, I suggest you use community advisory boards. And it doesn't matter whether the community is the community of St. Louis or the community of Parkinson's sufferers. You can bring these folks in as subject matter experts. They have a vested interest in research. They care. They're community members, and they have a vested interest in that community as well. So, how do you run a community advisory board? Well, I've been on them and I've helped run them, and I've learned a few things that I think are essential. So, to successfully use a community advisory board, the participants need the big picture. For them to invest emotionally, they need to understand what's going on. That can be a challenge with the acronym soup and with some of the things we face, but they need the big picture. The second, seek input on unsettled issues. If you're informing people about things that have happened and it relates to the big picture, that's good. But if you're just telling them things, or if you're seeking input and you're getting input on matters that aren't changeable, then everyone's going to become disconnected quickly because their efforts aren't germane and both sides will soon feel it. So number 2 and 3 are very closely related. We need to seek the input on the issues that they can still affect. And number 3, define tightly the boundaries of the feedback sought so that everyone has an understanding of what they can offer from their background and experiences that help answer the questions still at hand. The fourth is you actually need to develop procedures to make sure that all members get to speak. In person, there are people who talk a lot like me, but it's actually more challenging now that we're in a sort of hybrid environment, because if you are participating in a meeting by a cell phone, you are at a grave disadvantage compared to someone engaging from a desktop. And so we've found that we have to develop a deliberate process to make sure all get heard, or all will simply not be heard. And then this is the other corresponding point. Just as we're helping frame the big picture so people can understand and invest, but then seeking input on narrow issues, after that's done, seek open commentary to learn what you didn't know you needed to know. This lets them tell you things that aren't on your radar, and some of them could be actionable, but it also lets them be heard and bring up things. In the active studies, one of the things we talked about a lot was just how COVID was doing, what's going on with vaccines, what's going on with the numbers. I was representing rare disease participants there, but again and again, some of the other community advisory members had lost one family member, 2, and then another would be in the hospital. And these were important to hear about because they had experiences as community health workers and as people who had borne the brunt of the COVID pandemic. They had questions that were different. They had advice that was different, and it was really essential. So this is how we do it. This is how to balance these pieces to allow for big picture openness and broad commentary, but then also make sure that you've closely defined things so people can weigh in on what is actually able to be changed. If that happens, I suggest that the future of integrating patient input into research should be at least as bright as stuffed crust pizza's future. Thank you.
We have time for a couple of questions if there are any. I'm going to start you off.
Yes.
Because I think this has been, well,
There we go.
there are some. I'm going to start off the questions. I think this has been incredibly informative. And one of the goals we have with the current renewal of our ICTS is in fact to increase the community engagement at every level, including helping investigators have community advisory boards. And we have the support of our own community advisory board that they provide that educational framework. But you touched on one thing that I think might be worth the audience hearing your views on, because I've heard you talk about this, and that is the importance of relaying results.
Yes.
Do you want to talk a little bit about that?
Yes. So it's such a, when you're a participant in a study, you'd really like to know how it went. And getting those results back. I mean, we were involved in monthly meetings, and I'm excited to find out how active one turned out. And I'm at least, one thing I know is that learning something didn't help is also important. So, coaching participants up on the understanding that a result that was not a home run for a treatment identified still has real value, because you are part of a collected effort at a lot of research. And so, but returning the data to the people is just essential. And so I was giving a speech at Datapalooza, and I explained all the various things I did with my medical records and with literature. So I built a 363-page interactive PDF that was searchable and had links in it so that the doctors could get through my records, and it included literature. Because if I include the abstract, they're going to say, I need to see the whole paper. And if I give them the whole paper, they're going to say, I don't have time to read the whole paper. So I gave them both. And you could reach them and search them and everything else. I was able to show how having access to my imaging studies let me use my relationships with Vanderbilt, the NIH, Cleveland Clinic, U Chicago, that even, that my internist didn't have. And so we were able to get all of those folks to weigh in on those images because WashU was able to share the imaging with me. And so again and again, I walked through that. And at the end of the speech, I basically said, if you're one of the people who says, you don't know what a patient would do with their medical records, that's right, you don't. And it's the same with research. If you've invested and you've become a participant, you want to know how this made a difference. I mean, and especially with a situation like COVID, we were dealing with hospitalized patients. And it's powerful to know that the input you gave in the midst of your suffering informed something. And it's powerful for the families that remain. One of the challenges we saw with the genetic results is that sometimes a genetic condition might have been detected, and we had to figure out, do we tell them? It's not about COVID, but do we tell the participants? Do we tell survivors? How do we do that? And that's where the Community Advisory Board was very helpful in saying how they would feel, which is very different than maybe how the researchers would feel. And we got really impressive answers. I remember we had a discussion of a longitudinal study looking at Alzheimer's. And one of the Community Advisory Board members said, there are people who might not want to sit in a meeting and be told that based on their loved one's activities that they're looking in the mirror in the future. And so you may find a whole different level of resistance to participation than you would for other conditions. So these are the things that we get to learn early, rather than having to go through the entire process and get stalled out either at recruiting or find out that the relationship's been damaged even though the study went well. Because Cheryl and everyone else really wants the results and values those relationships.
So I don't see any questions. There weren't any questions. There don't appear to be any questions from the audience. Oh, wait a minute, here's one.
What's my favorite pizza?
So,
yes.
So Doug, what would your advice be to an investigator who wants to build a relationship with patient advocates but has no clue how to start?
Yeah. So I think that applies potentially to a lot of people, because you'd like to increase the impact of the work you're doing. And so I think that institutes like this are very helpful, because we have a community advisory board that is staffed with people who care about medical research and care about the community. And we are happy to try and facilitate relationships with people who come to us recommended by others who have a track record of good faith. So this institute has put us, apparently we're the only self-governing one. So Cheryl and I are doing extra homework, which is wonderful, but we have an opportunity. We have been pushed forward again and again with opportunities to participate, and that builds up a lot of trust. And so if researchers have guidance from people who have community input, and then they're able to be introduced to community partners that are already known to the university, I think that's the way to go. It may be a challenge to just show up out of the blue and say, I have limited social skills, but a great lab, and I'm here to ask you a lot of personal questions. But there are existing relationships that should be leveraged, and I think that's the way to go.
That might be an apt description of some of our colleagues. Anyway, but Doug, thank you very much. I want to thank you again for a wonderful presentation.
You're very welcome. Anytime.